We'll now move on to gastrointestinal physiology. And as we look at gastrointestinal physiology, we're going to have to approach this from several different angles. Imagine this as representing our GI tract with the stomach, the small intestine, and then the large intestine. What we'll see is that with the gastrointestinal system, we're going to see substances that are secreted by the GI tract itself into the lumen of the GI tract, and this may be from the lining of the intestine itself. It may also be from some accessory organs, and what we've shown here, it would be the substances from the liver as well as from the pancreas. We're also going to see that the GI tract itself will secrete substances that will go into the bloodstream. So that would be hormones, and these hormones can come back and will impact the physiology of the GI tract. And then, of course, the commonly known function is the fact that when we have substances moving through our GI tract, we will be digesting those and absorbing those substances. And those substances are going to be absorbed into a venous system, and this venous system then is going to be going straight to the liver. And it's primarily through a structure called the portal vein. So we'll be looking at all these different processes that go on in a stepwise fashion. So first, let's look at the anatomy of the GI tract and understand that in many cases, the morphology of the structures that are involved can help us understand what their functions are. So I'm going to bypass the oral cavity and pharynx and move straight to the esophagus. So the esophagus, this is a picture through a endoscope. So this is what's seen when a gastrointestinal physician will use their scope to look into the esophagus. So the esophagus is basically just this straight tube. And it's a tube that's lined by skin, and that skin is actually exactly the same as the skin that's on the outer surface of your body. The only difference is we don't have hair follicles. So this is not a setup which is designed to do any type of absorption. Purpose of the esophagus is going to be simply to transport material that we've eaten down into our stomach. This picture demonstrates this transition zone between the esophagus and stomach, and so we can see that there's a transition from this type of lining, which is the normal esophageal lining, to the lining of the stomach. So this is a perfectly normal appearance, and it gives us this sharp demarcation with regards to how things look. Now, one thing that we can talk about here at this point is the way that we move things along through the GI tract. So when we swallow food, food does not just simply move down by gravity. There actually are muscular contractions that are going to occur that's going to move the food through. So imagine these black lines represent the esophagus, and we have a what we call a bolus, which is just a small amount, a bolus of food that we have chewed and we've swallowed, and it's now entered the esophagus. In the esophagus, what's going to happen is there's going to be a sequential series of muscular contractions which are going to move that food from the top of the esophagus down into the stomach. These types of contractions actually occur throughout the GI tract, and these are called peristaltic contractions. We then will move into the stomach. And if we again take a look at an endoscopic view of the stomach, this is what the stomach looks like. So here as we look at this, it appears as though the stomach has lots of folds within it. And these are called rugae, R-U-G-A-E, but these are folds that are not there to provide an increase in surface area. This is just the way the stomach looks when it relaxes. As it's filled with food, what we'll see is that the stomach lining actually will flatten out. So this is what the surface of the stomach looks like. It has what we call pits. So the stomach lining itself is smooth, but there are these little openings, and these openings lead to gastric pits, and these are lined by different types of cells. And there are 4 cells that we're going to focus on in this class. The first are what we call neck cells, or mucous neck cells, and these are produced by carbonate. Another type of cell is called a parietal cell, and these secrete hydrochloric acid and intrinsic factor. intrinsic factor is necessary for us to reabsorb vitamin B12, which is important for making of erythrocytes. Another cell is called a chief cell, and these secrete a substance called pepsinogen and gastric lipase. And then finally we have our G cells, which secrete a substance called gastrin. So we'll talk about these different substances as we make our way through this lecture. From the stomach we go to the small intestine. In the small intestine we can divide into different segments. The duodenum is the initial portion. The jejunum is the next portion in line, and then the terminal portion is called the ileum. So I want to introduce these terms just so that you're familiar with them, but we don't need to know the exact demarcations of where one of these begins and the next one, or one of these ends and the next one begins. Now, in contrast to what we saw with the stomach, this is a picture of the small intestine. And here, these folds are actually built into the wall of the small intestine. So these are permanent. These are not things that are going to flatten as the small intestine gets filled with food. Although you have nothing to compare this with at this point, this lining of the intestine is actually very thick, very velvety. So this is a portion of our small intestine that we'll see has a morphology which makes it ideal for absorption of nutrients. So if we take a look at this, what makes up this lining, what we're going to see is that these folds here, this is what's represented by this drawing, these represent plica circularis. Now, if we'll zoom in on the surface of these plica, what we're going to see is that there are these little things that are jutting out. And these individually is called a villus. As a group, they're called villi. So this is a way that we can increase the surface area. Okay, so this represents one of these little areas here. Now we can zoom in on the villus more, and what we'll see that each of these individual areas here represents an individual cell. And on the surface of that cell, they have what we refer to as microvilli, and this is called the brush border. So we can see by this folding and the folding of the villi, the microvilli, this is a tremendous way to increase the surface area for absorption. Now, this is a good time for us to introduce another type of contraction that occurs within the GI tract. So we saw previously a peristaltic contraction, and its primary purpose was to move materials along, push them forward through the GI tract. Here at the small intestine, this is a good time to identify this type of contraction, which are called segmentation contractions. So imagine again that this is the small intestine in this case, and we have collections of food which are in the small intestine. Here what happens is the intestinal wall will contract, and as it contracts, it begins to pinch these areas of food in two, and then they contract a little bit differently. And each time they contract, they are mixing the contents of the small intestine. So these segmentation contractions, or segmental contractions, are ones which are not meant to move food forward, whether they are designed to mix the food with the contents of the GI tract, which will be important as far as digestion goes. We then move to the large intestine, or the colon is another word for that. You'll notice that the colon has this, what I've demonstrated here is just a red stripe. There's actually three of these on the outer surface of the colon, and this is a layer of muscle, and this is called the tinea coli, and this layer of muscle is very easily identified. We'll also see that the large intestine has these little scalloped appearance, and these are called haustra. So for a surgeon, these are very easy to be identified so that we can tell the small intestine from the large intestine, even if we have a relatively limited view. You'll notice that we have this little structure dangling off. This is the appendix. The first part of the colon is called the cecum. We then can identify these other structures here by which direction they're running. So this is called the ascending colon, the transverse colon, the descending colon, and then this S-shaped area we call the sigmoid colon. This is a picture, an endoscopic picture showing inside of the colon, and this area here is where the ileum is going to be dumping into the cecum. So this would be right here. So this is called the ileocecal valve. Now we can look at the lining of the large intestine, and here we're going to see that this appears much different than the small intestine. So this lining relative to the small intestine is very thin, very transparent almost appearing. So this is not a lining which is set up to do a lot of absorption. At the end here we have the rectum, and the opening is called the anus. Now if we look microscopically at what this surface of the lining of the large intestine looks like, very much like the stomach in that it's flat. And it does have these pits. Here though, these cells that are in these pits are primarily mucus producing cells. So in the large intestine, we're not seeing any significant amount of absorption other than some water. We will be absorbing water here, and that will help to compact the feces. But by the time the contents have reached the large intestine, all of our nutrients have been absorbed. Another picture here of the large intestine, and we can kind of get a hint of the haustra, the little scalloped appearance that we see on the outside. We can also see to some degree here on the inside as well.